Welcome to the deep dive. Our mission today is, well, highly specific, very scientific, and it's focused entirely on the microscopic engines that power every single second of your life.
That's right.
We've been handed a stack of intensely specialized biological and anatomical texts. These focus on the absolute machinery of existence, the cell, its powerhouse, and the incredibly complex, regulated system that supplies the fuel.
Yeah, and these sources, they don't just offer descriptions, they're more like the operating manual.
Yeah.
They quantify and detail the how and the why behind cellular function, behind the energy systems. It gives us a really unprecedented look into human metabolism right down at the molecular level.
And you, our listener, you're looking for the deepest possible explanation of this relationship between cellular energy, especially the mitochondria, and the metabolic pathways that regulate, well, everything. This is really the ultimate under-the-hood deep dive into how your body sustains life. We're going from the smallest structural unit all the way up to complex hormonal command and control.
And to do that, we really have to anchor ourselves firmly in molecular biology and energy thermodynamics. We'll start by defining the fundamental unit, the cell. Our sources confirm it exists at the fifth level of human organization.
Right, bigger than organelles, smaller than tissues.
Exactly. Superior to organelles, inferior to tissues. Then we'll rapidly zoom in on the specific architecture of the powerhouse, the mitochondria, to understand those core energetic processes that sustain every single function, from, the blink of an eye to the secretion of hormones.
Okay, let's unpack this then. Starting with the basics that literally hold everything together, the foundational architecture of the cell itself. The sources are absolutely clear on this. The cell is the fundamental, morphological, functional, and genetic unit of living matter. It really is the basic cornerstone of biology. Every complex action we take, everything you do, starts with the structure and function contained within that microscopic boundary.
And it doesn't matter what their specialization is. Could be a nerve cell, a muscle cell, a gland cell. They all share the same fundamental structural plan. It's built on three main components.
Okay.
You've got the protective outer boundary, the cell membrane, sometimes called the plasmalemma. Then there's the internal operating environment, the cytoplasm. And finally, the command center, the nucleus.
Let's focus first on that boundary layer, the membrane. Because it's not just a sac holding things in, is it? It's this dynamic gatekeeper defined by the famous fluid mosaic model. What does that model actually tell us about how the membrane manages the cell's environment?
Well, it's a really elegant design. The structure is mainly composed of phospholipids and various types of proteins. Now, the phospholipids, they naturally organize themselves into a double layer that's the lipid bilayer. And here's the genius part. The heads of these phospholipids are hydrophilic. They love water, so they face outwards, towards the watery environment, both inside and outside the cell. But the tails, they're hydrophobic. They repel water, and they face inward, creating this lipid core that's sandwiched between the two surfaces.
And that hydrophobic core, that water-repelling center, that's the key to its integrity, isn't it?
Absolutely. That sort of greasy, non-polar core. It acts as a highly restrictive barrier. It fundamentally prevents the easy, passive passage of most water-soluble molecules, and critically, ions like sodium or potassium.
Which is vital.
Oh, completely vital. If the cell couldn't control the flow of ions, its electrical potential, its osmotic balance, it would all just collapse instantly.
Okay, so if that core is basically a hard stop for anything dissolved in water, how does the cell get nutrients in or get waste out?
That's where the mosaic part of the model comes in. Embedded within this continuous lipid structure are all these various proteins. You've got transport proteins, receptor proteins, enzymatic proteins, and they aren't uniformly distributed. They're dotted around, hence the mosaic appearance.
Like tiles in a floor.
Kind of, yeah. And these proteins, they are the selective gates and the pumps. They handle all the specialized functions, like active transport, where energy is used to pump things against their gradient, or facilitated diffusion, where they help specific molecules across. They ensure that only specific needed substances like glucose or particular amino acids are moved bidirectionally across this barrier, and crucially in a controlled way.
So the membrane isn't just a wall, it's a functional powerhouse in its own right, separating the inside from the outside, the extracellular fluid.
Precisely. It maintains that crucial internal environment.
Okay, so moving inward then. Once we get past that very selective membrane, we enter the cytoplasm. The sources describe this as a complex colloidal system. Sounds like the cell's internal factory floor.
It really is the factory floor, and it's dynamically organized. The cytoplasm itself is composed of two main parts. There's the non-structured fluid matrix, we can call that the hyaloplasm.
That jelly-like stuff?
Essentially, yes. Water is the dispersion medium, suspending colloidal micelles. And then you have the structured part, which includes all the specialized internal compartments, the organelles. This whole environment supports the incredibly complex chemical reactions that sustain life.
And among these organelles, the sources really highlight the machines responsible for, well, building complex structures and breaking down spent materials. Let's look at the primary synthesis centers first.
Right, so we begin with the ribosomes. These tiny structures, they're rich in ribonuclear proteins, and they are the absolute assembly line for life. They are the principal universal site of protein synthesis. They read the genetic instructions, like mRNA, to link amino acids together into functional proteins.
And their operational environment, where they often work, is defined by the endoplasmic reticulum, or ER.
Exactly. The ER is like the cell's internal circulatory system and processing plant. It's a network of tubules and flattened sacs called cisternae, linking the outer cell membrane sometimes all the way to the nuclear membrane. And we distinguish between two forms based on whether they have ribosomes attached.
The rough and the smooth ER.
Correct. The rough ER, sometimes historically called the ergastoplasma, it's studded with ribosomes, giving it that rough appearance. Its main job is synthesizing and modifying proteins that are destined either for secretion out of the cell or for insertion into the cell's own membranes.
Okay.
Meanwhile, the smooth ER, it lacks ribosomes, hence smooth. It's critical for a whole different set of processes like synthesizing lipids, steroids, detoxifying certain chemicals, and importantly for our discussion, glycogen metabolism. It manages the storage through glycogenesis and the release, glycogenolysis, of glucose reserves right there within the cell itself.
So, rough ER makes proteins for export or for membranes. Smooth ER handles lipids, detox, and those internal energy reserves. But like any complex factory, you absolutely need a cleanup crew, handling waste, broken parts, maybe even foreign invaders.
And that's the absolutely critical role of the lysosomes. These organelles are basically small sacs filled with powerful hydrolytic enzymes. Think of them as the cell's recycling center and waste disposal unit.
What kind of things do they break down?
Their job is intracellular digestion. They break down everything from obsolete organelles that are past their prime, damaged proteins that aren't folded correctly, to particles that the cell might engulf from the outside, like bacteria, or even fragments of tissue. This is why they are particularly abundant and very active in phagocytic cells, cells like macrophages and certain white blood cells that specialize in cellular housekeeping and defense.
Okay, so we have the blueprint from the nucleus, the perimeter control with the membrane, the internal transport system of the ER, the assembly lines with the ribosomes, and the cleanup crew with the lysosomes. But all these processes, building things, moving things, cleaning things up, they require massive amounts of energy.
Huge amounts.
The nucleus might control the genetic template and oversee metabolism, but it delegates the actual heavy lifting of energy production. And this brings us straight to the organelle that serves as the universal battery charger, where the sheer volume of energy conversion actually happens.
Indeed. You know, it doesn't matter how perfect the instructions are coming out of the nucleus or how efficient a ribosome is at folding a protein. If the currency of energy, which is ATP, adenosine triphosphate, is not constantly regenerated, the cell dies almost instantaneously.
Wow, that fast.
Seconds, maybe minutes at most for some functions. This vital function is reserved almost entirely for the mitochondria.
All right, here's where it gets really interesting because the architecture of this specific organelle, the mitochondrion, it seems perfectly engineered for just massive energy throughput. Let's zoom in. What is its unique structure that allows it to capture what the sources say 95% of the energy from our fuel?
It's quite remarkable. Mitochondria are typically oval or spherical in shape, and they're surrounded by a trilaminar lipoprotein wall, essentially a double membrane system. There's an outer membrane, which is relatively smooth and quite permeable to small molecules.
Okay.
But then there's the inner membrane. This is where the magic happens. This inner membrane is highly specialized, and it's thrown into numerous deep folds. These folds are called mitochondrial cristae.
And the space enclosed by this folded inner membrane is called the mitochondrial matrix.
Okay, those folds of cristae. Why are they so critically important? Is it purely about packing more membrane into a small space? It's much more than just saving space, although it does that too. The function is fundamentally, inextricably linked to that folded structure. The mitochondria are the site where the later stages of fuel breakdown happen. The Krebs cycle occurs in the matrix. But most importantly, it's the site of oxidative phosphorylation.
That's the main energy-releasing process.
That's the big one. This is the process that releases the vast majority of energy stored in our food molecules and captures it in the form of ATP synthesis. And this incredibly complex chain of enzymatic reactions involving proteins embedded in the membrane, collectively called the electron transport chain, happens directly on the surface of those cristae.
So the cristae aren't just dividing walls. They're literally the reaction surfaces themselves.
Yeah.
How does that folding translate into energy capture efficiency? It seems key.
It's absolutely key. The folding massively maximizes the surface area available. Think about it, if you just had a smooth inner sphere, you'd have far less room. The folds allow for the insertion of thousands upon thousands of protein complexes, including the critical enzyme ATP synthase, which are necessary for high-efficiency energy production.
How does it work? How does that surface area help make ATP?
Well, the electron transport chain, using energy from breaking down fuel, pumps hydrogen ions, protons, from the matrix across the inner membrane into the small space between the inner and outer membranes. This builds up a massive electrochemical gradient, essentially a high concentration of protons in that inner membrane space. It's like charging a battery.
Okay, a proton gradient.
Exactly, a proton gradient. And the vast majority of the ATP generated from oxidizing glucose, we're talking about 34 or so molecules per glucose, are generated when those protons flow back down their concentration gradient, rushing back into the matrix through specialized channels. These channels are part of the ATP synthase complexes, which are also embedded in the crista.
So the flow of protons turns something.
It literally turns a molecular rotor within the ATP synthase enzyme. This rotation drives the phosphorylation of ADP, adenosine diphosphate, plus a phosphate group into the high-energy ATP molecule. Without those folds, you simply couldn't physically house enough of these electron transport chains and ATP synthase machines to meet the cell's energy demands. Life, as we know it, wouldn't be possible.
The scale of that production is just phenomenal, and the sources detail how ATP is that currency, the universal energy link. It stores a significant amount of energy. The figure given is around 12,000 calories per mole in its two high-energy or macroergic phosphate bonds, ready for immediate use anywhere in the cell. But the sources also introduce a different molecule, one that holds even more energy per molecule. That seems counterintuitive, maybe. If ATP is the working currency, why have something else with more punch?
That's where the true genius of cellular engineering reveals itself again. It comes down to the need for speed and an immediate high-power reserve. That other molecule is phosphocreatine, often abbreviated as PC or sometimes CP.
Phosphocreatine.
Yes. And it holds an even higher energetic value per molecule, roughly 13,000 calories per mole, slightly more than ATP. And critically, it's often several times more abundant than ATP within the cell, particularly in tissues with very high and fluctuating energy demands, like skeletal muscle or brain tissue.
Okay, several times more abundant and slightly higher energy. So the obvious question is, if PC has more energy, why doesn't the cell just use it directly for muscle contraction or pumping ions? Why bother with ATP?
It's because PC isn't engineered to interface directly with the vast majority of cellular machinery that actually consumes energy. Myosin heads in muscle, ion pumps in membranes, they are all specifically designed to bind and hydrolyze ATP. Phosphocreatine's role is strictly specialized.
What is that role then?
It acts as an immediate, high-capacity, high-speed buffer or reserve. Think of it as a rapid recharge station designed solely to maintain stable ATP levels during sudden bursts of high-intensity activity.
So like an emergency generator for ATP.
Kind of, but even faster. Imagine a muscle fiber starting to contract powerfully, maybe during a sprint or lifting a heavy weight. The ATP levels inside that cell will dip instantly as it's consumed.
Right.
Almost simultaneously, an enzyme called creatine kinase catalyzes the reaction. PC plus ADP rapidly converts to creatine plus ATP. This reaction provides an instantaneous boost, regenerating ATP almost as fast as it's used up in those first few critical seconds.
And that's faster than the mitochondria can ramp up.
Much faster. It takes several seconds, maybe even longer, for the whole mitochondrial oxidative phosphorylation process to accelerate fully to meet a sudden, massive increase in demand. The phosphocreatine system bridges that gap.
So it's the ultimate cellular emergency battery system. It doesn't directly power the headlight, let's say, but it instantly recharges the battery that does.
That's a perfect analogy. It provides an immediate, high-capacity reserve system supporting sudden, powerful energy demands. This system is absolutely crucial because while the aerobic system, oxidative phosphorylation in the mitochondria, provides the highest yield of ATP over time, The PC system provides the necessary power output, the rate of ATP supply, in those first few seconds of maximal exertion.
The difference between a quick sprint and a slow jog really comes down to that initial burst powered by PC.
To a large extent, yes. The efficiency and capacity of that phosphocreatine reserve is critical for high-intensity, short-duration performance.
Okay, so to keep those mitochondria churning out ATP and to keep that phosphocreatine system topped up, the cell needs fuel and a coordinated way to process it. Our sources lay out the three main fuel sources, glucides, which are carbohydrates like glucose, lipids or fats, and proteins. The body manages these with incredible, precise pathways. Let's start with the immediate preferred energy source, glucose.
Right. Glucose is the fast-access fuel. It's readily available and relatively easy to metabolize. The journey begins right there in the cytoplasm with a process called glycolysis. This literally means sugar splitting. It breaks down one molecule of glucose, which is a 6-carbon sugar, into 2 molecules of a smaller 3-carbon compound called pyruvic acid, or pyruvate.
And this initial step, glycolysis, it happens outside the mitochondria.
Yes, entirely in the cytoplasm.
Hmm.
And it releases only a tiny fraction of the total potential energy stored in that glucose molecule. But the crucial fork in the metabolic road occurs right after glycolysis, and it depends entirely on whether oxygen is available to the cell.
And the sources really stress the monumental difference in efficiency based on that oxygen presence. It's huge.
Oh, the difference is night and day. Aerobic efficiency, meaning with oxygen, is the goal. When oxygen is present, that pyruvic acid produced by glycolysis is actively transported into the mitochondria. Inside the mitochondrial matrix, it's further processed, enters the Krebs cycle, also known as the citric acid cycle, and ultimately feeds electrons carried by molecules like NADH and FADH2 into that electron transport chain located on the cristae that we just discussed.
And that's where the big ATP payoff happens.
That's where the vast majority happens. This highly efficient oxidative phosphorylation process yields approximately 95% of the total ATP extractable from that glucose molecule. We're talking about typically around 34 additional molecules of ATP generated inside the mitochondria, compared to just 2 net ATP from glycolysis itself.
What's fascinating, too, is the built-in energy demand sensor you mentioned, the self-regulation. How does the cell know when to stop making ATP?
Yeah, it's remarkably elegant. The process is inherently self-regulating based on the energy status of the cell, specifically the ratio of ATP to ADP. Key substrates required for the Krebs cycle and oxidative phosphorylation include ADP and inorganic phosphate. When the cell is using energy rapidly, ATP gets broken down to ADP and pi. High levels of ADP signal low energy, and this stimulates the whole process to run faster.
Makes sense.
Conversely, when energy demand is low, ATP levels build up and ADP levels drop. Once essentially all the available ADP is converted back into ATP, the enzymatic machinery of the electron transport chain and ATP synthase effectively stalls. There's simply no ADP left to phosphorylate. The presence of ADP signals energy depletion and high demand. The absence of ADP signals energy sufficiency, and the engine idles down almost instantly. This prevents the wasteful breakdown of fuel when ATP reserves are already full.
Very efficient. But what happens when the oxygen supply fails? Say, during really intense exercise where the muscles outstrip their oxygen delivery, or maybe temporary circulatory impairment. The cell shifts to what the sources call a desperate, inefficient salvage operation.
That's the anaerobic emergency pathway, anaerobic meaning without oxygen. Anaerobic glycolysis, which is basically just glycolysis running without the subsequent mitochondrial steps, is indeed extremely inefficient. As you noted, it yields only about 3% of the total potential energy from glucose, just those 2 net ATP molecules per glucose.
So why rely on 3%? I mean, if it yields so low energy, what makes it worth the metabolic trouble? Why even have this pathway?
That's a great question. The tiny energy yield isn't the primary immediate benefit here. It's actually about cofactor regeneration.
Right.
During glycolysis, a crucial step requires a molecule called NAD+ to accept electrons, becoming NADH. Now, in aerobic conditions, that NADH travels into the mitochondria and unloads its electrons to the electron transport chain, regenerating NAD+ in the process. But without oxygen, the electron transport chain isn't running. So NADH would just build up and NAD+ would run out. If NAD+ runs out, glycolysis itself would grind to a halt. No more glycolysis, no more 2-ATP, cell death.
Okay. So how does anaerobic glycolysis solve that NAD+ problem?
The pyruvic acid produced at the end of glycolysis, instead of going into the mitochondria, is immediately converted into lactic acid. This conversion step uses up the NADH generated earlier in glycolysis, oxidizing it back into NAD+. This regeneration of NAD+ is absolutely vital because it allows those initial energy-yielding steps of glycolysis to continue running, even without oxygen.
So the conversion to lactic acid isn't about the lactic acid itself, it's about getting NAD plus back so glycolysis can keep sputtering along.
Precisely. That tiny 3% ATP yield from continued glycolysis is all that's keeping the cell alive, buying it precious minutes until oxygen returns. Once oxygen is available again, the accumulated lactic acid can be cleared, either converted back to pyruvic acid and used aerobically, or transported to the liver to be converted back into glucose.
So it's really a structural maintenance mechanism under duress, more than a significant energy producer.
Okay, now, to manage the immediate glucose supply, the body stores it. The sources mention glycogenesis mainly in the liver and muscles.
That's right. Glycogen is basically a branched polymer of glucose molecules, like a storage depot. The liver stores glycogen primarily to maintain blood glucose levels for the whole body, especially the brain. Muscles store glycogen for their own use during activity. Together, these stores provide a readily accessible reserve of roughly 3,000 kilocalories.
Which sounds like a lot, but it's only enough for relatively short duration efforts, maybe an hour or two of moderate exercise.
Exactly. It's a short-term buffer. And if those circulating blood glucose levels, what we call glycemia, start to drop below the normal range, which is very tightly regulated between about 65 and 110 milligrams per deciliter.
A remarkably narrow range. Incredibly narrow, yes. If glycemia drops, the body absolutely must restore it. This triggers gluconeogenesis, which literally means making new glucose. This process, primarily in the liver, synthesizes glucose from non-carbohydrate precursors, mainly certain amino acids derived from protein breakdown, lactic acid recycled from anaerobic metabolism, and glycerol from fat breakdown. This ensures the brain, which relies very heavily, almost exclusively on glucose, remains constantly fueled.
Okay, so once that relatively small 3,000 kcal reserve of fast fuel glycogen starts running low, or if we're talking about longer-term energy needs, the system then turns to the high-density reserves that really define long-term survival, lipid metabolism or fats.
And this is where the sheer scale of the body's energy storage becomes truly apparent. Lipids serve multiple vital functions, of course, but their energy role is just monumental. They are the principal energy reservoir in the body. When fats are broken down, oxidized, they release about 9.3 kilocalories per gram.
Compare that to glucose.
Compared to glucose or protein, both of which yield around 4.1 kilocalories per gram. So fat provides more than double the energy per unit mass. And because we can store large amounts of fat in adipose tissue, a normal, healthy individual carries an enormous lipid energy reserve. The sources estimate roughly 50,000 kilocalories, maybe even more.
50,000 kilocalories from fat versus maybe 3,000 from glycogen. That's a staggering difference. It really underscores how the human body is fundamentally engineered for endurance and long-term survival, primarily through fat storage.
Absolutely. But beyond just energy, lipids perform other essential structural and functional roles. Their plastic role, as the sources call it, means they're indispensable structural components of all cell membranes. Phospholipids, cholesterol, they form that fundamental bilayer. Lecithin is mentioned as an example.
And the functional role?
Their functional role is critical in endocrinology, the study of hormones. Cholesterol, which is a type of lipid derived either from the diet or synthesized in the body, serves as the essential precursor molecule for synthesizing all the vital steroid hormones. This includes the glucocorticoids like cortisol, the mineralocorticoids like aldosterone, which regulate salt balance, and all the sex hormones, estrogen, progesterone, testosterone.
So fats are not just fuel, they're structure and signaling molecules too. Now, the sources detail a really unique digestive and absorption path for lipids that's fundamentally different from how we handle glucose or amino acids. They can't just be absorbed directly into the bloodstream from the gut, can they? Why the special treatment?
It all comes down to their chemistry. Fats are hydrophobic, they don't mix well with water, and both the contents of our gut and our bloodstream are primarily aqueous, water-based. So first, dietary fats, mostly triglycerides, arriving in the small intestine need help. They require bile salts produced by the liver and stored in the gallbladder, along with phospholipids like lecithin to act as detergents.
Emulsification.
Exactly, emulsification. They break down large fat globules into tiny microscopic droplets, vastly increasing the surface area available for digestive enzymes called lipases to work on them. These enzymes then hydrolyze the triglycerides into fatty acids and monoglycerides.
Okay, so they're broken down, then what?
Then, these breakdown products, along with cholesterol and phospholipids, are absorbed into the cells lining the intestine, the epithelial cells. But they can't just pass into the blood from there. A crucial repackaging step occurs inside these intestinal cells. The triglycerides are reassembled and then combined with specific proteins, cholesterol, and phospholipids to form large lipoprotein particles. These particles are called chylomicrons.
Chylomicrons. And these are too large for standard transport into the blood capillaries.
Exactly, they're quite large. Because of their size and their lipid-rich nature, they cannot pass directly into the small blood capillaries within the intestinal villi the way simple sugars and amino acids do, which go straight to the liver via the portal vein. Instead, these chylomicrons must enter the lymphatic circulation.
The lymph system. Why there?
They enter specialized lymphatic vessels called the central lacteals, which are present in the core of each intestinal villus. This specialized transport pathway via the lymph system collects the chylomicrons, which then travel through larger lymphatic ducts and eventually get released into the general blood circulation, typically near the heart via the thoracic duct. This route effectively bypasses the immediate first-pass processing by the liver that glucose and amino acids undergo.
That's a fascinatingly complex detour. It really highlights the challenge the body faces in handling these high-density but water-insoluble fuel sources.
It does. It's a specialized system for a specialized fuel.
Okay, finally, the third category of fuel, the building blocks themselves, which are typically only used for energy in extreme circumstances. Protein metabolism?
Right. Proteins make up a huge portion of our body mass, nearly a quarter, the sources note. And their primary metabolic goal isn't energy production, it's structural maintenance and growth, what we call anabolism. Building and repairing tissues, enzymes, antibodies, transport molecules, all of that.
Where do the amino acids, the building blocks for this, come from?
They come from three main pools. First, directly from the protein in our diet. Second, the body can actually synthesize some amino acids, the non-essential ones, from glucidic or lipid precursors if needed. And third, there's a constant turnover, a necessary breakdown, catabolism, of existing body proteins to recycle amino acids and remove damaged or unnecessary proteins.
So, if proteins are primarily the structural components in functional machinery, when does the body actually resort to using this vital infrastructure as fuel?
Protein degradation, primarily for energy, which also yields that same 4.1 kilocalories per gram as carbohydrates, is typically the absolute last resort. It really only becomes significant when the massive lipid stores and the smaller glycogen reserves are severely depleted. Think prolonged starvation or certain disease states.
And the sources mention something about phases of depletion during starvation.
Yes, it's quite interesting. They describe protein depletion occurring in roughly 3 distinct phases during prolonged starvation. There's an initial rapid breakdown phase as easily accessible proteins are used. Then there's a second, much slower phase where the body tries hard to conserve its vital protein structures. And finally, if starvation continues, there's a third, terminal phase characterized by rapid, massive muscle wasting and breakdown of functional proteins just before systemic collapse occurs.
Wow, so that final phase represents the body literally dismantling its own structural foundation, its muscles, potentially even organ proteins, just to feed the absolute minimal energy demands of the brain and other vital organs.
It's a desperate survival mechanism, using the building blocks themselves as the final fuel source.
Okay, so we have this vast interconnected cellular energy system, glycolysis, the Krebs cycle, oxidative phosphorylation, fat breakdown, protein use as a last resort. This cannot possibly run unsupervised. It has to be tightly controlled. It requires an orchestra of hormonal signals responding instantly to the body's needs, to the environment. We already mentioned the incredibly tight maintenance of blood glucose, glycemia, which ranges only from about 65 to 110 milligrams per deciliter. Maintaining that narrow window seems like it would require flawless, continuous feedback and control.
That narrow range is the definition of metabolic homeostasis, keeping things stable. And the endocrine system, the system of hormones, is indeed the master regulator. It operates primarily through a beautiful system of antagonistic or opposing hormones that fine-tune fuel availability and utilization. The pancreas is really the frontline commander when it comes to glucose management.
Right. Let's elaborate on that great antagonistic pair from the pancreas. They seem central.
They are absolutely central. On one side, you have insulin, which is secreted by the beta cells located in the islets of Langerhans within the pancreas. Insulin is the key hypoglycemic agent. Its main job is to lower blood sugar levels when they rise, typically after a meal.
How does it do that?
It acts as the signal of nutrient abundance. It facilitates the entry of glucose from the blood into most body cells, especially muscle and fat cells, by increasing the number of glucose transporters on their surface. It promotes the immediate utilization of glucose for energy. And crucially, it stimulates the conversion of excess glucose into glycogen for storage that's glycogenesis primarily in the liver and muscles.
And its effect on fat.
Very important. In lipid metabolism, insulin actively suppresses the breakdown of stored fats, lipolysis, and vigorously stimulates the creation and storage of new fat, lipogenesis. As our sources point out, this action provides the precise physiological explanation for why consuming excess carbohydrates, more than needed for immediate energy or glycogen storage, inevitably leads to fat deposition. Insulin basically directs the body into storage mode for all fuel types.
Okay, so insulin lowers blood sugar and promotes storage. What's the opposing force that prevents blood sugar from dropping too low, say between meals or during fasting?
That is glucagon, which is secreted by the neighboring alpha cells, also in the pancreatic islets. Glucagon is the primary hyperglycemic agent. Its job is to raise blood sugar levels when they fall too low. It acts as the signal of nutrient deficit or need.
And its actions are the mirror image of insulin's.
Essentially, yes. Its main action is to stimulate the breakdown of stored glycogen in the liver, glycogenolysis, releasing glucose into the bloodstream. And, very importantly, it also strongly stimulates gluconeogenesis in the liver, the creation of new glucose from amino acids and other non-carbohydrate precursors. Glucagon basically dictates, release stored fuel, make new fuel, get blood glucose levels back up immediately.
So insulin and glucagon provide that minute-to-minute balancing act for blood sugar. But beyond this immediate pancreatic balance, other hormones step in, especially when the body faces a more prolonged challenge or a significant stress state. The glucocorticoids, with cortisol being the main one in humans, seem particularly complex in their role.
They are complex, yes, because they orchestrate a kind of metabolic reallocation strategy, often associated with chronic stress or longer-term adaptation. Their complexity stems from this dual strategic role in fuel management. Glucocorticoids, like cortisol, strongly stimulate gluconeogenesis in the liver to ensure a continuous, even elevated supply of blood sugar.
Where do they get the precursors for that new glucose?
This is the strategic part. To provide the building blocks, amino acids for gluconeogenesis, cortisol promotes catabolism or breakdown of proteins, particularly in skeletal muscle. It essentially tells muscle tissue to release amino acids. But simultaneously, it promotes anabolism or building up in the liver, where those amino acids are immediately snapped up and used to synthesize not only new glucose, but also essential liver proteins.
So it's breaking down muscle to support the liver and maintain blood sugar.
In simple terms, yes. It's a strategic dismantling of less immediately vital peripheral tissue, muscle, to support more vital central functions, liver function and brain glucose supply, during prolonged stress. Cortisol also exerts a potent lipolytic effect, mobilizing fatty acids from adipose tissue deposits, providing an alternative non-glucose fuel source for many tissues, thus sparing glucose for the brain.
So cortisol's message is kind of like, okay, team, we might be in this for the long haul. Let's sacrifice some muscle mass to keep blood sugar high for the brain, and let's start burning more fat.
That's a good way to capture the essence of its metabolic effects. And then contrasting with that more prolonged strategic response, we have the hormones that trigger an instantaneous massive energy mobilization. The catecholamines, adrenaline, also called epinephrine, and noradrenaline, or epinephrine.
The fight-or-flight hormones.
Yeah.
These are secreted during the most acute forms of stress, right? Trauma, fear, extreme cold.
Exactly. They're secreted heavily by the medullary part of the or adrenal gland during sudden, intense stress, physical trauma, extreme temperatures, life-threatening situations, even profound emotional strain, and they act instantly and powerfully across multiple systems.
What are their immediate metabolic effects? It sounds like they just dump fuel into the system.
That's pretty much it. Metabolically, they trigger immediate widespread breakdown of glycogen, glycogenolysis, in both the liver and muscles, leading to intense acute hyperglycemia, a rapid spike in blood sugar to fuel immediate action. They also cause rapid mobilization of fats from adipose tissue, including promoting the immediate catabolism or burning of fatty acids for energy.
And the sources mention a non-metabolic effect too.
Yes, critically, these hormones don't just mobilize physiological energy, they also produce profound effects on the brain, causing cortical alertness, heightened awareness, and often anxiety. This directly links the sudden dumping of fuel reserves into the bloodstream with the necessary mental state for rapid reaction and survival in a perceived emergency.
So it's fuel plus focus, instantly. Okay, finally, we have the hormones that seem to regulate the general metabolic thermostat, the baseline rate at which all our cells are burning energy.
That's primarily the influence of the thyroid hormones, T3 and T4. They act more broadly and chronically to increase the body's overall metabolic rate, essentially the general rate of energy utilization or heat production across nearly every cell type in the body. They turn up the furnace, so to speak.
And how does that connect to fuel mobilization?
Well, this systemic increase in energy burn naturally requires a corresponding increase in fuel supply to sustain it. So, thyroid hormones indirectly lead to the rapid mobilization and utilization of fats from storage depots simply because the overall energy demand of the body is elevated. The entire energy expenditure system is measured in calories, or more usually kilocalories, and the sources note that high sympathetic nervous system stimulation, which releases adrenaline, and high levels of thyroid hormones are primary factors in dramatically increasing that overall metabolic rate.
So the ultimate purpose of this incredibly complex web, the cellular machinery, the fuel pathways, the antagonistic hormonal signals, the stress responses, as we've seen throughout, the maintenance of that stable internal environment, keeping things like blood glucose, temperature, pH within very tight limits despite huge variations in the external environment, our activity levels, and what we eat. That concept of homeostasis.
Precisely. Homeostasis is the goal, and this intricate interplay of energy metabolism and its regulation is absolute fundamental to achieving it. Hashtag outro.
Wow. That was a truly detailed and specific deep dive into the microscopic engines that keep us, well, keep you running. We move from the incredible selective barrier of the fluid mosaic model membrane all the way into the specialized folded architecture of the mitochondria, and finally detailed that complex hormonally regulated flow of glucose, high-density fats, and, as a last resort, critical proteins that feed that cellular engine.
Yeah, when you look at all the source material together, what really stands out is the sheer, almost unbelievable engineering brilliance required for life. That staggering 95% energy yield difference between aerobic metabolism and anaerobic glycolysis hinges entirely on the structural integrity and the vast folded surface area of those mitochondrial cristae and, of course, the availability of oxygen.
That's incredible.
And then to realize that this entire high-performance system is instantly, continuously managed by antagonistic hormones like insulin and glucagon, working tirelessly to keep your blood glucose within that critically narrow 65 to 110 milligram per deciliter range. It's truly remarkable.
I was also really struck by that detail about phosphocreatine. It's not just a high-density storage molecule, technically holding slightly more energy per mole than ATP.
Right, 13,000 calories versus 12,000.
But its role is purely as this ultra-fast supercapacitor, engineered only for rapid energy transfer. It's useless for doing the actual cellular work itself, but it's absolutely indispensable for instantly recharging the working ATP currency during those first few seconds of intense effort. It really demonstrates that cellular speed, the rate of energy delivery, is just as vital as the total energy capacity.
Absolutely. Power versus capacity. And this complex interplay, especially thinking about the link between those acute stress hormones, the catecholamines, and their massive metabolic fuel mobilization effects, leads us to a final provocative thought for you, our listener, to consider. We know that adrenaline and noradrenaline evolved to mobilize enormous energy reserves and stimulate cortical alertness in response to immediate, severe physical threats or trauma, the classic fight or flight. Given that these same hormonal mechanisms are triggered, perhaps at lower levels but more chronically, by the psychological stresses and anxieties of modern life, how profoundly does this constant low-level alert signal tax your body's metabolic reserves compared to actual short-term physical exertion? What might be the hidden, compounding physiological cost to homeostasis when the body is continuously signaled to dump fuel reserves into the bloodstream, often without the corresponding physical activity to actually burn them off?